A couple of videos back, we talked about the structure of DNA and went in depth on the different bonds that take place on it. This time, I wish to touch on how DNA gets replicated. It all begins with a wound chromosome, specifically euchromatin. Unlike its brother heterochromatin, euchromatin is loose and able to be expressed or replicated. This makes it easy for DNA to be unwound for replication. After getting selected, helicase breaks the previously discussed hydrogen bonds between nitrogenous bases and opens a place for DNA polymerase to work. Primase now picks a nucleotide and starts the process. It does this by placing Okazaki fragments onto the chosen nucleotide that later gets switched to a DNA nucleotide. This enzyme only occurs in a 3 to 5 direction, and the Okazaki fragments are made from RNA. The phrase 3 to 5 comes from the deoxyribose sugar on the DNA backbone. The order of carbons goes clockwise from the oxygen at the point of the sugar, from 1 to the 5th being the one that connects to the phosphate. Other than the 5th and the 3rd carbon, there are certainly other memorable carbons. The 2nd carbon is where the lone hydrogen that differentiates DNA from RNA in the absence of oxygen is located, the deoxy part of DNA. Although I didn't draw it here, the 1st carbon is what connects to the nitrogenous base. When these sugars get ordered to form the backbone, the leading strand originally follows the 3 to 5 pattern and is continuous, while the lagging strand is the one on the bottom that follows the 5 to 3 pattern. In replication, the starting DNA turns into 2 DNAs. The other strands follow the opposite of their original pattern, changing from 3 to 5 into 5 to 3 and vice versa. The varying strands are what cause DNA replication to be described as antiparallel or semi-discontinuous. Sometimes you may see people say that the leading strand runs from 5 to 3. This is because the complementary strand that the nucleotides get added on runs from 5 to 3. If you are still confused by this, please watch my next video. All in all, this breaks the replication phase into 2 strands, the leading and the lagging. Each of them produce their own complementary strand, eventually splitting off. This does it for this video. There are a lot of details to go into, and I am aware that people's attention spans on this app aren't that long, mine included. I will be returning shortly with the second half of the DNA replication.